Hello, everyone, and welcome to today's webinar, Connecting the Dynamics of Antimicrobial Response Across Scales. I'm Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Molecular Machines and Industries. To learn more, visit www.molecular-machines.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box located in the lower left of your screen and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you're experiencing any technical issues, you can use this Q&A box and we will assist you directly. Lastly, there are interactive poll questions after the presentation. We do appreciate your participation in our polls and we'll let you know when to submit your responses. I'd like to now welcome our speaker, Dr. Somenath Bakshi, Associate Professor, Department of Engineering at the University of Cambridge. Dr. Bakshi, you may now begin your presentation.
Hi, everyone. First of all, thank you for the kind introduction. It's a pleasure to present our work on trying to connect the dynamics of antimicrobial response across scales, which we think is quite crucial to develop a predictive understanding of antimicrobial response. And what we mean by response across scales is as follows. So in our lab, we have developed experimental methodologies to analyze the heterogeneity of antimicrobial response at the level of individual cells. To understand the origin of this heterogeneity, we analyze the underlying gene regulatory mechanisms to see what genes are contributing to the spectrum of response that we study across a population. And to understand the consequence of this heterogeneity, we use computational models and experiments to predict and test the population dynamics of response. So today, my talk, here's a brief outline of it, where we'll first discuss the motivations of this work and the associated challenges. We will then describe the method developments that we have done to be able to address those challenges. And then we'll move on to analyze a set of problems to illustrate what kind of systems we can study using such an approach and what we have learned from those. And I will end with a discussion of future directions of this work. Now, in terms of motivations, the biggest motivation, of course, is the alarming spread of antimicrobial resistance, which refers to the phenomena in which bacteria that were previously treatable with antibiotics becomes untreatable due to the fact that they acquire some genetic factors which make them resistant towards antibiotic treatment. Now, the magnitude of this problem is quite serious. So last year, it was predicted or estimated that about 1.3 million people died from resistant infections. And the prediction is that by the year 2050, annually 10 million people will die if it continues at the current rate. So it's an important problem that we need to tackle. And in order to contribute to the global effort towards combating this alarming spread of antimicrobial response, my lab is contributing in two different directions. In one, we are trying to understand the factors that underlie the emergence and spread of AMR. And on the other hand, we're trying to develop and test strategies that are alternative to antibiotics so that we can spread the stop the spread of these resistant infections. Now, the approach that we are taking is grounded at the level of physiological context of individual cells. The reason being is that often the mechanism of antibiotic action is well understood at the level of molecules. So how an antibiotic binds to its target. And it is also quite straightforward to quantify the response and resistance evolution towards antibiotic treatment at the level of population. But in order to connect these two scales, we must understand what is going on at the level of individual cell. Because to develop a mechanistic understanding of how a population responds and eventually becomes resistant towards treatment, we need to really understand the physiological context of the problem, because it is the physiology of those individual cells that determine whether the drug can get inside the bacteria, can it reach the target, or does the cell pump it out. It also determines whether there are sufficient number of targets available in the bacteria and whether they are active or not, so that the drug can actually bind them. And also, the physiology determines what is the fitness trade-off of mutating the gene of the target site, which shapes the resistance evolution. However, this sort of physiological picture cannot be studied from a bulk measurement because individual cells are quite different from each other. One of the underlying reason is stochastic gene expression, but there are various others. To illustrate this problem, Michael Lilloig's lab did an excellent experiment where they used 2 copies of the same gene and expressed a red and a green fluorescent protein from them. And instead of cells appearing yellow, different cells appeared in different colors, suggesting that there is a lot of variability in the level of individual gene expression. Because if they were expressed at the same level, you'd expect every cell to have same amount of red and green, so they would appear as yellow cells. Now, this heterogeneity has quite important significance in response dynamics at the level of population. So, for example, I'll refer to the case of antibiotic persistence, which is a scenario where within a population of growing cells, there are these rare red cells that are dormant, and as a result, they are refractory towards antibiotic treatment. So when the antibiotic treatment is going on, they don't respond, but once the antibiotic is removed, these cells revert back to the normal stage and they recreate the original population. So antibiotic persistence is a serious problem because it underlies recurrent infections, because the entire population cannot be eliminated. Now, not only population response, the evolution of a population is also often determined by this heterogeneity. In a beautiful experiment done by Natalie Balaban's lab, it was shown that the lag time of cells from exiting dormancy is a feature that determines their tolerance towards antibiotic. And if populations are evolved against different durations of antibiotic treatment, so here 3 hours, 5 hours, and 8 hours, the distribution evolves to sort of adapt towards the treatment durations. So together, I think these examples clearly illustrate that in order to understand the origin of this heterogeneity and its consequence towards resistance, evolution, and response towards treatment, we must understand these processes at the level of individual cells. And so then only we can bridge this mechanistic sort of understanding to develop a predictive understanding. And so that's the approach my lab takes. And so here are 3 systems or problems that we're trying to analyze using this approach. So first, we're trying to understand how does the physiological heterogeneity and environment of bacterial cells affect their response and resistance towards antibiotic treatment. Then one of the alternative antimicrobial treatments we are looking at is using actually viruses called bacteriophages, which can specifically target bacteria and eliminate them. And there we are asking similar questions because phages also, as a parasite, depend on bacteria's physiology. So we are asking how does the physiological heterogeneity of the bacterial cell affects the kinetics of phage infection and the coevolution of the 2 systems. And finally, we look at the outliers of the spectrum of this physiological heterogeneity, which are often rare events such as persister cells, events that lead to loss of plasmids which carry antibiotic resistance, and their transfers between different cells, and how these phenomena affect the recurrent infections and spread of resistance. Now, of course, we understand that in order to study these processes, we have to carry out single cell measurements to characterize the cellular heterogeneity. But we must first understand what are the requirements for those single cell measurements so that to be able to quantify the heterogeneity reliably. So we think that sort of single cell measurements has to contain dynamic information and information about cell morphology. So FACS is quite a well-used platform or methodology that enables people to look at a really large number of cells over time and snapshot them. But while it comes with a very high throughput, it does not contain any image. And in case of bacteria, the backscattering has poor information about the cell size. And also, we don't get any dynamics from these sort of measurements. Snapshot imaging using various techniques can actually provide the additional information about cell size and morphology, give more information about the response towards antibiotics, but there is no tracking. And so we can't really quantify the response dynamics and the fluctuation in this heterogeneity over time. To address these challenges, a technique was developed which was both convenient but also quite rich in data, which is to track individual cells as they are seeded in this agar pad containing growth media, and these cells will then grow and divide to form these microcolonies, enabling us to track their growth dynamics and gene expression. So time-resolved single-cell imaging of bacteria in growing microcolonies has been a very well-used technique. But this, while it being a convenient technique, it actually comes with severe artifacts and limitations that we have recently uncovered. So one of them is that when you seed a bacteria on an agar pad and it goes to form these colonies, the environment actually starts to change as a result of this growth. So if we look at the amount of nutrient at the center of a colony, it goes down over time, whereas at the periphery, the nutrient stays flat, which means the profile of nutrient across these cells is not homogeneous. So we can't reliably say that the heterogeneity that we're analyzing is the intrinsic heterogeneity, because it might be a result of the environmental heterogeneity that you have imposed on the system. But even if we could somewhat eliminate that problem or mitigate it, it would be very difficult to quantify the heterogeneity. And this sort of results from the diffraction limited nature of our imaging system. So here I will introduce the system, the point spread function, which is the diffraction limited sort of nature of microscopic imaging. So if you image a single point, rather than it being imaged as a point, it gets imaged as this blurry point spread function. As a result of this, if you have a cell containing 100% of the emitters, when you image it, only 94% of those cells, the emitters, photons actually stay within the cell. The others are sort of assigned to pixels outside the bacteria. And this is sort of a perfect model of a point spread function. In reality, these effects are actually much, much worse. As a result of this point spread function, we get a new problem, which we have termed as point spread function bleed through. If you have a single cell, so here is a schematic where you have this cell with all the emitters in this gray area. As a result of the point spread function, the light gets out of the boundary. And if you have two cells next to each other, some of the light from one cell goes to the cell next to it and vice versa. This is what we term as bleed through effect. Now, if we run simulations, we have checked that an individual isolated cell is always dimmer compared to cells that are part of larger microcolonies, is because their neighbors contribute a lot of these intensities through bleed through. And the effect is quite serious, as you can see that here is an experiment we have run to verify this process. And we see that isolated cell plotted in green here, they are of the order of 2 to 3 times dimmer than cells that are part of larger colonies, the orange data here. So the massive heterogeneity that one sort of sees in the total data, the blue data here, is actually a result of just different colony size, not so much about the actual heterogeneity of the individual cells, right? And so suggests that both the platform's lack of homogeneous environment and the imaging system's inability to pick up this underlying heterogeneity makes it an unreliable platform to quantify this process. So there are other sort of nuanced effects as well. I'll briefly mention it, is that it also makes it very hard to quantify heterogeneity's direction. So if you have no heterogeneity, the structured aspect of the microcolony imposes a heterogeneity. So CV, which stands for coefficient of variation, is, if we use a simulation where the ground truth is zero, you actually get a 12% variation just from the result of this bleed through effect. On the other hand, if you had a high variability, so here the ground truth was 35% variation, as a result of the bleed through, which act actually as a smoothing function because it takes intensity away from the bright cell, it reduces the variability to 12%. So if you observe a variability of 12%, you don't know whether it actually came from sort of a low variability situation or a high variability situation, which makes it very difficult to computationally correct. So instead of trying to find ways to sort of alleviate that problem, we actually go for a very different solution using microfluidics. So this is a platform called Mother Machine, where individual cells are isolated and maintained in their own trenches, right? And so because the cell at the bottom produces its progenies, it is termed as Mother Machine because we term that as the mother cell. And in this device, we have an array of these very narrow trenches where a single line of bacteria can grow, and they are fed from media in an orthogonal channel which diffuses through these trenches, and as a result there is no competition between bacteria and no depletion of nutrient. So this is a sort of a movie of such a platform, and you can see the cells are growing continuously over time, and they are growing in a spatio-temporal uniform environment, which is essential for quantifying the physiological heterogeneity. Furthermore, because of the structured nature of the device, we can actually space them apart to ensure that there is no point spread function bleed through. So not only we can ensure that the cells are in uniform growth condition, we can actually also ensure that we can quantify the heterogeneity reliably. And we have done recently a lot of optimization of these device designs using various lengths and widths to ensure that we can get the right kind of environment and retention to carry out the necessary measurements. And so using this device, you can then track individual cells as they keep growing and dividing and producing their progenies. And we can do that for hundreds of generations. And from this data, we can actually then quantify how often cells are dividing, how fast are they growing, and what is the dynamics of gene expression of a certain reporter within them. And we have made this platform now very high throughput. So the current throughput of the system is 132,000 cell lineages. So we are not anymore limited by the amount of data that we can collect and so here actually we have a full montage of the entire of the device, and a single field of view is shown here, and a smaller crop of that shows you 4 lineages. So in total, we can track 132,000 cells, which provides us the ability to get really good statistics, but also to detect rare events. Now, this imposed a challenge with data analysis. So as I mentioned that we look at cells and then quantify gene expression dynamics using a fluorescent reporter. So to quantify the growth dynamics and also the gene expression dynamics, what we need to do is we need to isolate these individual trenches, which looks like this. And then within these trenches, we have cells that are sort of dimmer than the both the media background and the device background. And then we have to identify their boundaries, overlay them on the fluorescence channel to quantify exactly how much intensity there is. And also from the shape of the boundaries, we can estimate the size of these bacteria and their growth rates over time. Now, that's not an easy task. It's because the point spread function in this bright field is almost as large or of the similar order of magnitude in dimension as the bacteria that we're trying to analyze, right? So here are some example images of bacteria that we use. And as a result, if we have to do machine learning analysis of this data, because automated analysis or manual analysis, of course, is not a option, because we're collecting of the order of 10 to the power 7 cells per experiment. So we need automation. But if we try to do machine learning, we can't generate reliable training data for these sort of images, because human beings, we can't really identify exactly where the cell ends and begins, because the blurring function is so large. So using human annotators to accurately annotate data and generate training data is not an option. So in order to tackle the challenge of not having the right kind of training data, we developed a alternative approach which uses a virtual microscopy pipeline called SIMBAC, which stands for synthetic microscopy images of bacteria, where we can actually incorporate the accurate model of cell growth and division in the microfluidic environment, their physical interactions with each other and the device itself. And from these simulated data, we can extract ground truth images that are now perfect for the corresponding image. And we generate the image using the phase contrast microscope model that represents the optics that we use. What this enables us to do is that we can rapidly generate large amount of synthetic data that are very realistic. So if you compare with the panel below, that's real image of bacteria in our microfluidic device. But the data, synthetic data comes with perfect ground truth, so, which means we can now train machine learning models that can look at real data and produce very accurate masks. So I'll present some example performance, and you can see on the left, these are real cells growing in the microfluidic device, and the machine learning model trained with SIMBAC data is producing very accurate outlines of the bacteria. Now, SIMBAC, first of all, makes the job for an annotator much easier because nobody has to then sit down and do it, but it is also very, very fast. So it's about a factor of 1,000 times faster than a human annotator. So here are some timing benchmark. But not only it's efficient and user friendly, it actually makes the process of generating data quite accurate. And because we now had synthetic data with perfect ground truth, we could actually benchmark human annotators. And we found that human annotators, because of the small size of bacteria, typically underestimate the size of their boundaries. And if we make the cells even smaller by putting them in stationary phase, their extent of underestimation goes even down. And this results from a combination of two effects. One is that images are projected images in wide field microscopy, and the projection makes it difficult to know exactly where the cell begins and ends. And the second effect is diffraction, as I have explained before. And these inaccuracies in ground truth data translates into inaccurate performance of the machine learning models trained with them. And so if we look at the delta model, which is one of the popular model for image segmentation in bacteria, you can see the performance is quite erratic, whereas if we take the same model and retrain it with SIMBAC data, we get a very precise analysis of the cell width, which is the shortest dimension, so actually shows the inaccuracies much more clearly. And not only it improves precision, we have now in a recent paper we have shown that it also improves accuracy. So as you can see, the purple data here are from SIMBAC, and for various sort of image conditions, it retains the accuracy of the segmented cells. And it's quite a versatile platform, meaning that you can generate images for different types of bacteria. So here's some E. coli cells, there's bacillus, then bacteria under different growth conditions where they get smaller and start to stack differently in different imaging conditions. So different types of microscope objectives and also different imaging modalities, like going from phase contrast to fluorescence. And it is also versatile towards different imaging platforms. So we can use the microfluidic device mother machine, as I mentioned. Then there are microfluidic chemostats where patches of bacteria are imaged in a single layer. And in all of these situations, SIMBAC can generate realistic data that can be used to train models and produce very accurate segmentation results. So now that we have established that with the optimized microfluidic platform and machine learning models trained with virtual microscopy synthetic training data, we can actually accurately analyze physiological heterogeneity and heterogeneous response dynamics. Let us sort of look at an example of how this sort of approach can be applied to analyze response towards phage treatments. Now, phages are these viruses that adsorb to target bacteria, inject their genome, and reproduce them, sort of replicate themselves, and then eventually lyse the bacteria to reinfect other bacteria in the culture. Now, each of these process or the steps in infection are critically dependent on the physiology of the bacteria. And as we have seen that because the physiology of the bacteria is quite heterogeneous in a population, we do expect that the kinetics of this process is going to be heterogeneous as well. As a result, bulk measurements are not going to be able to deliver or give us the kind of information about these underlying dynamics, because they are going to blur these dynamics by averaging over asynchronous events on cells with variable physiology. So we have developed a method to carry out these measurements at the level of individual cells so that we can quantify the timing and variability And not only that, because we are imaging these individual cells, we can link the extent of variability to the physiological variability of the infected cell population. So we actually developed a different version of mother machine to study this process of phage infection at the level of individual cell. Now, in this mother machine, we have these very shallow 400 nanometer deep trenches through which the phage can diffuse and infect bacteria. Because we found that in the regular mother machine, phages would immediately absorb to the cell at the bottom of the trench and get washed away, where we couldn't actually study the process in until completion. To analyze this process, we used a phage called T7, which our collaborator Fusco Lab engineered in a way that it contained a YP reporter in its genome, so that every time a phage infected a bacteria, the bacteria would glow in yellow. And that was enough for us to optimize the microfluidic design. So we compared the regular mother machine designs with a narrow site range and the design with a wide site range, and found that only in the wide site trenches we could get 100% infections and clearance. So we proceeded with that. So I will show you now, and which probably is the first time you'll see phage infection at the level of individual cells in a sort of a time result data. So here's a movie, and if I start the movie, I'll just quickly stop it right here. So you're seeing a field of view with 9 trenches where cells are loaded and they were undergoing exponential growth continuously. And you'll see when a phage swims up and infects a bacteria, it will initially turn yellow, then it will pop and release viruses, which will go to infect all of the other bacteria in the trench, and they will disappear over time. Because all of these cells are undergoing exponential growth until the point of phage infection, we can assume that the physiological heterogeneity is unchanged, and we can truly see the effect of infection at the level of individual cells. So here you can see, if you pay attention to the trench on the right, that cell becomes yellow, and then you can see the clearance is happening now in every cell. And if you pay attention to this trench, an infection here happens at the bottom and gets washed away. And as a result, this trench never gets cleared. So I will replay this movie and you can follow it again. That trench suggests that even though every trench is producing thousands of phages, they're not cross-contaminating each other. So these are all independent effects where we can study phage-induced growth halt, lysis, and successive rounds of lysis in the trenches. So these events are very fast because phages take only 18 minutes, this particular phage T7, to infect and lyse a bacteria. And so we had to collect the data at 30-second time resolution. So I'll play one of those trenches with a bit slower time scale so that we can follow the process. So if you see this particular bacteria, it's about to get infected. So it starts to glow in yellow because it expresses that capsid reporter. And then it pops, produces phages, which go to infect all the other cells in the trench. They all start to glow yellow, and then they stochastically just pop over time. And so we can look at each of these trench to produce this chymograph, which shows you which cell was infected, and when it disappeared, how the other cells in the population got infected, and how they disappeared over time. And using the machine learning model described before, we can actually track each of these infection events and quantify how cells were growing and dividing before infection, then upon phage infection, how their growth slows down and stops, and then right after that, how the phage produces its capsid protein and lyses the bacteria. We can quantify various time scales from this data, the time at which the growth arrest happens of the host, the production of the capsid reporter starts, the cell lyses away, and we can quantify the heterogeneity across the population to see exactly how these time scales are distributed across individual cells and the time gap between different events. Interestingly, we found that if we look at the cell length data from which we can quantify the growth rate of individual cells, then we can see that for various cells at different stages and different lengths, when the phage infected them, their growth rate followed a very reliable pattern as it sort of eventually stopped. But the production of phage proteins was quite variable across the population and had about an order of magnitude difference between the sort of minimum and the maximum level of production. Not only that, very interestingly, we found that this variability was explained by the length of the infected bacteria, because the correlation factor between the length of the infected bacteria and the final intensity was pretty high. And so here they're all colored by their length of infection, suggesting that the observed heterogeneity is indeed coupled with the physiological heterogeneity of the host. Now, so far we have focused on the steps after the host was shut down and then phage proteins are produced. But we can actually soak the phage using a DNA binding dye so that its own genome gets stained by this dye, so that we can follow exactly when a phage lands on the bacteria and injects its genome. So here I will show you one of those examples. So if you pay attention to this cell, you'll see that orange particle just landed and it immediately, its signal disappeared because it injected its genome into the bacteria. And this particular cell is now doomed. So all the other cells are continuing growth, but this one is just disappears. So if I go back to this time, right here, I think we can see that yellow particle appearing, the orange particle. And so if we plot the orange signal, we see that's the time when the particular phage arrived. And so if you look at the high resolution picture in shorter time scales, we see that the signal gradually decayed over time. And so from this decay, we can actually quantify the kinetics of genome injection by that single phage. And a second phage actually came, but by that time the bacteria was already infected, so it couldn't inject its own genome. And so this one sort of disappeared after a certain point. And from this data, we can quantify the adsorption to injection times across infection event. You can see that it's quite variable. The mean injection time is about 5.6 minutes. Put together, what we have now is that we have a new assay that can enable us to study multiple steps of the infection happening at the level of within the individual cells, from the point of adsorption, genome injection, host takeover, expression of capsid protein, and eventual lysis. So it's the first time we can actually quantify the kinetics of this process and associated variability across infected cells. This provides us with a assay which we can use to link to physiological factors that underlie this variability. But also, because this assay, the device itself is quite scalable, it enables us to screen different types of engineered and natural phages in terms of their kinetic properties to identify really effective ones. Now, finally, I'll sort of get into the discussion about how can we have now observed that we can quantify the dynamics of antimicrobial response or phage response at individual cell level, but how are we going to connect it to the population level? And this was motivated by also a need for us to look at dynamically changing environment, because in nature, often environments are sort of changing over time because food arrives, then population of bacteria consumes them, they go into sort of starvation period until food arrives again. And in laboratory, we simulate them by growing cells in a culture flask. So they multiply over time, the food runs out and cell density goes up. And our question was like, how can we analyze this process at the single cell level? So we implemented a very simple idea, which is hook up a microfluidic device, the mother machine, with the flask. Now, the cells in the culture are depleting its media, and so this depleted media will flow through the device, and the cells in the mother machine will respond to this depleted media as if they themselves are also in the flask. So we're treating the mother machine as an array to trap bacteria which are immersed in the culture. Now, while it's sort of a simple enough idea, implementation was very tricky because you have to couple a bulk assay, shaking culture, with microscopy, and had to measure several challenges of flowing a dense culture through microfluidic devices. But now this sort of platform works, and I can show you here at the bottom, we are looking at population dynamics of the culture. So it's optical density of cells in exponential phase at the beginning very low, and then they grow up. And on the top, you get single cell response. So the stress response is shown in the fluorescence level, and the growth of the bacteria you could see from their size and their rate of change in size. So as soon as the media is replenished, the cells get out of the stress stage and they become large. Their stress level goes down, and they are growing and dividing rapidly until the food runs out again, and then they slowly start to stop dividing and growing, and the stress level goes up. So this is the first time we can quantify this sort of response at the level of individual cell, while we can also monitor the population dynamics. Now, from this data, we can quantify various features. So what is the stress response dynamics across the population? So in the orange data, we're plotting the response from the master regulator of stress response, the RPUS level. In the blue data plots the cell size and growth over time, and in the black data plots the population density. So we can track exactly how physiological spectrum changes as a function of their position along this changing cell density in the culture class. And I will point out here that you can see that these are not related to features that are heritable. So for example, you can see the orange trace was from a cell that had a very high stress response in the first round of stationary phase, where the optical density was also high, the black data. But in the second round, it went to a low stress response level. On the other hand, the red trace is from a cell that had a low stress response in the first round, but had a very high stress response in the second round. So shows that these are caused by physiological heterogeneity and stochastic decisions, not something that is genetically caused by genetic differences. Same thing is true for the dynamics of cell length changes over time. Now, to ensure that indeed what we are seeing in the mother machine is what's going on in the flask, we actually took snapshots of the cells in the culture flask, and we found that the two types of data match or follow each other very well. So in blue solid line, we have the continuous data from mother machine, and the dots are from the snapshots. And you can see that the cell length as the culture is going into stationary phase follows really nicely in both methods. And the same is true for the stress response level. And we also look at exit dynamics, and again we see that the population really follows from the culture really follows the dynamics measured using the mother machine. But the mother machine is providing this continuous data, so we can actually analyze physiological heterogeneity and fluctuations over time. So the data from tracking single cells as they enter and exit stationary phase actually shows a very interesting pattern. We find that some cells, like the blue one here, continues to divide until the very end of stationary phase, and as a result they become very small. And when the cells are provided with fresh food, then these cells wake up much later compared to the red cell, which actually decides to divide much earlier. And it stays large, and as a result actually wakes up much earlier when fresh food is provided. This trade-off, however, cancels out because we see that if we look at the number of division by a bacteria during exit and during entry and during exit, we see that the total actually is constant, irrespective of whether it's a small cell that divided more often during entry and less in the exit, and or if it's a large cell which divided more during exit but less during the entry, right? Because we see that the total is fixed. So in typical stationary phase, like an overnight culture, this strategy of diversifying ways of entering stationary phase does not seem to have a benefit. But if we look at a much longer, like a 72-hour long, 3-day long stationary phase, we find that only the larger cells are able to wake up. So this is the largest 10%, which shows that almost 80% of them can wake up, whereas the smallest 10% do not wake up at all if we are provided with fresh media after 72 hours. And the probability of waking up is monotonically dependent on the size of the bacteria. So suggesting that if cells don't divide during stationary phase, they actually have an advantage when they wake up. So what would be the point of staying small in stationary phase? Because you're wasting sort of the chance of a revival. But instead of providing fresh food, if we actually treat the population with an antibiotic, we find that it is actually the small cells that can survive the antibiotic treatment by laying dormant. And when the antibiotic is removed, then these small cells go to recreate the population. And these are the persisters. So you can see in these traces here that the gray cells, the larger ones, wake up very quickly when the fresh food and antibiotic is provided. And so they are killed. But the orange ones live to sort of just stay dormant during the treatment, and eventually they wake up to recreate the original population. And so when we looked at about 20,000 cells, more than 20,000 cells, we found only 200 cells that were small were able to persist the antibiotic treatment. Now, put together that now we can say that we have this ability to connect dynamics of microbes antimicrobial response across scales, because we can use a bioreactor and the coupled inline density sensor to monitor its optical density over time to see how the population is behaving. And we can hook up the microfluidic device, the mother machine, to look at the single cell dynamics of these cells as we treat them with an antibiotic. Not only that, this bioreactor can then be hooked with a robotic arm that can deposit the samples over time into a 96 well plate, which is maintained using a Peltier cooler at 4 degrees centigrade, so that we can take a detailed fossil record of the long-term dynamics of this population, which we can study using a barcoding method to see their phenotypes over time, and also we can sequence them. So these combinations of method enables us to connect the response across scales. We can do high throughput single cell tracking, link them with the population dynamics over short time scale and over long time scale to understand the evolutionary dynamics. Now, where are we going with these methods also? Like, what's the next step? So one of the things that we can actually harness this high throughput for is to do high multiplex measurements. And because we want to know how to analyze these underlying genetic factors behind this observed physiology. So the method that we have developed in our lab uses a combination of dyes that have very specific colors and spatial distribution to label an entire library of knockout collection. Then we load them in the mother machine, and the computer vision method then identifies which cell came from which well based on their color and spatial distribution of the signals, right? And the remarkable thing about the mother machine device is that once we identify from the first snapshot what is the identity or what is the position of the mother cell, which well it came from, then it is the progenies of that cell which takes over that well. So even if the dye washes away, the identity of that particular genotype remains. So this enables us to do pre-screening genotype identification to map the phenotype to genotypes for the observed dynamics. But often we don't have a structured library like this. Often maybe we're interested in looking at a natural collection of isolates or mutants. In such scenarios, we want to use an optical tweezer to carry out the measurement where we load the collection of bacteria in the mother machine, carry out the time-lapse measurement, and then use the optical tweezer to grab cells of interest and take them for sequencing. So here is a movie of such a process done by our collaborator Johan Paulsson's lab, where they're grabbing that individual cell using an optical tweezer and then depositing it in a separate trench where the cells are sent for sequencing. And this is something we are also now establishing in our lab. Now, the work by Luro et al. Was done manually, which meant that it was very low throughput. Cells had to be collected one by one. And so this is why we have bought this MMI cell manipulator system, because they bring the capability of having multiple optical tweezers which can operate in parallel so that we can automate this entire process. So with that, I will end my presentation and just summarize the key lessons that we think we have learned so far. I think we have established that virtual microscopy platforms such as SIMBAC are going to be very useful to identify artifacts in image formation and also to design experiments and analytical pipeline to mitigate those. These also help us to design proper microfluidic devices that can address the challenges identified from the simulations. And microfluidic devices are also going to be quite an important sort of tool for this type of analysis. The synthetic data generated from virtual microscopy pipelines are very useful because we can use them to train machine learning model, which boost their performance. And together, these tools enable us to do time resolved measurements at the level of individual cells to uncover really interesting physiological factors that were not observable at the level of bulk measurements. And by connecting single cell and population dynamics using the microcosm platform that I have described before, we can understand how this physiological heterogeneity influences population fitness and evolution. And this we can now bring at the level of sort of screening multiple cells to really do high throughput and highly multiplex measurements to identify what are the genotypes and regulatory factor that underline the observed physiological dynamics. With that, I will thank the people that are involved in this work. I have an excellent team of PhD students in my lab and a postdoc postdoctoral research assistant and supported by 2 excellent lab technicians. Also, thanks to my collaborator and the funding agencies, and also the excellent environment in Cambridge that supports us and provides the inspiration of doing some of this work. And I will thank you for your attention and happy to take any question.
Thank you, Dr. Bakshi, for your informative presentation. Before we begin our live Q&A session, we do have a couple of poll questions as a reminder. So you should now see the first poll question on your screen. It asks, would you like us to provide you with additional information about the MMI technologies for cell manipulation and single cell isolation? If you can just please select yes or no, we would greatly appreciate the poll responses. We'll give you a couple of seconds for that one. And again, you should see the poll up in the middle of your screen where the slides were for the presentation. If you can just select yes or no to this poll question. And then we'll move along to our second question before Q&A. All right, moving on to our second poll question here. This one asks, are you familiar with the use of optical tweezers technology in single cell measurements? If you could select one of the following, not at all, somewhat familiar, very familiar, or expert, and go ahead and click submit when you have your response. I see your responses coming in, so I'll give a couple more seconds here. Again, we do appreciate your participation in these poll questions. All right. Thank you all for your participation in that poll session. Now we're going to dive into the live Q&A portion of this webinar. If you have questions you'd like to ask Dr. Bakshi, please do so now. Just go ahead and put them into the Ask a Question box located in the lower left corner of your screen, and we'll get to as many questions as we have time for. Our first question here, Dr. Bakshi, asks, how do rare outliers within this heterogeneity influence antimicrobial response and population survival?
Yeah, that's a very good question. Thank you. So the particular rare outlier that I focused in this particular talk is called antibiotic persistent bacteria. So these are cells that enter like randomly, like stochastically into dormant state. And in this dormant state, the active molecular targets that antibiotics target are actually, they're not active anymore. And as a result, antibiotics don't work on them. So these small cells therefore ensure that a very, very tiny population actually survives the treatment. And because these are stochastic processes that lead to entering into this dormancy and exiting out of the dormant state, when the antibiotic is removed, these cells can get back to the sort of original growing population and can recreate the population again. So you get recurrent infections because of them. So they are the sort of rare population that ensures that your population does not go to extinction and can always come back to restart the infection. Hope that answers your question.
Yes, thank you. All right, our next question here asks, does microbe diversity contribute equally to resistance of both antibiotics and phages?
Yes, the second part of the question. So for the first part of the question, it is definitely true that we have a good experimental evidence now that diversity definitely contributes to resistance evolution. So for example, there are sort of similar to persisters, there are also populations that are called perseverant population. So these populations of cells can continue to grow slowly during antibiotic treatment. These are small populations, but as a result of their continued growth and division and DNA replication during the treatment process, they can sort of help resistant mutants to emerge during treatment. Now, this kind of analysis is less or much less explored in the scenario of phage infection. But because phages depend on cell physiology in quite a similar way as many antibiotics, we would expect that indeed population heterogeneity would contribute to resistance evolution. Now, computationally, we have explored this problem in our lab, where we have looked at that if we have heterogeneous response in terms of timing of lysis towards a infecting phage, then sort of by shifting the lysis time distribution, you can have different sort of treatment outcome. But experimentally, it's something that remains to be explored. And so I think with our platform, we are well positioned to be able to answer that question in a more concrete manner in future.
Wonderful. Thank you.
Mhm.
All right. Our next question here asks, have you used your technology to look at different growth dynamics comparing intracellular and extracellular bacteria?
Intracellular bacteria, is it referring to cells, bacterial cells that grow in mammalian cells? If that is the case, then no. We have so far looked at only sort of bacterial cells by themselves. They're either in these monolayer microfluidic devices that we have in the lab, or they are growing in suspension culture. But we haven't looked at bacteria that are intracellular, like growing um, within another cell, like a mammalian host, if that's what you're asking.
Wonderful. And if that wasn't what they were asking, I'm sure they will send a follow-up question. Our
Okay.
next question here asks, how does cell-to-cell heterogeneity in bacterial populations affect their response and recovery from antimicrobial treatment?
So one of them, I think I mentioned that one of them is the antibiotic persister, because within a population you have different levels of metabolism, and some of these metabolic outliers have so little metabolic energy that they are refractory to treatment, and so they make the population does not go to extinct and cause recurrent infection. There is another class of sort of cells that actually have extra enzymes beyond what the antibiotic can titrate out, and those sort of extra enzymes can actually allow them to continue growth during the antibiotic treatment. So we call them the perseverant population, and so this population can subpopulation can continue growth, and so during sort of MIC level treatment, you will actually see that the optical density does not remain flat. There is a continuous growth due to the small population, and then they affect resistant evolution as well, because these cells are allowed to grow while the treatment is being done.
Okay, great. Thank you. Our
Mhm.
next question here asks, single cell measurements are necessary to quantify cell-to-cell heterogeneity in biological systems. But what are the necessary requirements for accurate quantification of this heterogeneity?
Yeah, that's a good point. I guess I tried to cover that also in my talk, is that of course it is now well understood that if you do bulk measurements, the mean and the standard deviation between measurements is not going to tell you about the underlying heterogeneity, and so there is a lot of motivation for doing single cell measurements. But as we showed in our talk today, that just doing single cell measurements doesn't mean that you're actually able to accurately quantify the underlying heterogeneity. So the two critical requirements that we focused on today are: one, you have to ensure that the environment is spatiotemporally homogeneous, so that you can compare the physiological heterogeneity across the population and over time, to be able to really compute this sort of to really be able to make sure that you are studying the intrinsic heterogeneity, not something that is a result of the environment where you have placed your system in. And so that's one necessity, where sort of microfluidic space systems which enable you to control the environment very precisely are a good way to start. The second aspect is that once you ensure that indeed the environment is spatiotemporally homogeneous, so that you can quantify physiological fluctuations accurately, you now have to ensure that your measurements don't have the artifact. They do allow you to quantify the heterogeneity that is the ground truth of the process. And that's where sort of designing the imaging platform in a way that imaging artifacts such as point spread function bled through, which leads to crosstalk of intensities between cells, are eliminated or mitigated to the point that they have very minimal effect. And also machine learning models that take into account of these diffraction limited images, so that you can quantify cell size and their growth dynamics accurately, are very much necessary. And so both sort of designing your image collection platform and your image analysis platform in a way that they ensure that you are able to study and quantify the heterogeneity accurately are necessary. Just doing single cell measurements or being able to isolate them is definitely not enough.
Okay, wonderful. Thank you.
Yes.
We have time for just a couple more questions here, so we'll keep moving along. Thank you all for your interesting questions. Our next one asks, does this diversity empower the bacterium regardless of resistance mechanisms?
Possibly not. I think definitely diversity is a context dependency phenomena, right? And so as you can see that phages, for example, they are parasitic and they can actually leverage this diversity to become more efficient. So if the host becomes diverse in a way that gives advantage to their parasite, then it's not good for bacteria. And similarly, there are support sort of context in antibiotic treatment where we think that diversity can lead to have more fitness cost than the benefit it gives for towards antibiotic resistance. And so it's a very sort of a context dependent phenomena. But these outlier events, like these very very rare events, are typically advantageous. Like for example, persister cells, because they don't have much fitness cost on the population, and also like horizontal gene transfer that leads to transfer of resistant genes to other species present in the same system, or sort of their own species that don't have that gene. So these are advantages. But similar rare events, for example, plasmid loss can actually lead the population to lose the resistant genes. So those can also become problematic. So these diversities can go both ways. So it's a very context dependent sort of a phenomena where they have advantages or disadvantages.
Okay, great.
Yeah.
Our next question here asks, can the laser capture technology isolate persister bacterial cells, then use these cells for downstream mammalian cell infections or other assays?
Yeah, definitely. That's definitely can be done. And that's one of the things that we are working on in our lab right now, is to use the optical tweezer to isolate these persister cells. Our primary target is to be able to do RNA-seq to kind of identify what is the gene expression profile. Most of the time we're using them for doing DNA sequencing, because one limitation right now is that the number of cell you collect per unit volume of liquid is typically a bit low to be able to do this kind of RNA-seq or follow-up assays. DNA sequencing is pretty easy because you kind of start from a single cell that you have isolated and you grow them up. So as long as your mammalian cells are also in the same device, so that you can drag the persister cell and infect them with that individual cell, then it's going to work. But if you need any sort of sample amplification, then it becomes a bit tricky, because as soon as that cell needs to grow, it will exit out of the persister state. So
Great.
that's just a limitation to be aware of. Yeah.
Okay, thank you. And we have time for just one more question. That question asks, how does physiological heterogeneity among bacterial cells affect their predation by bacteriophages?
Yeah, so that kind of is a very nice question. It's the answer is also a bit nuanced, but I'll try to sort of give you a bit picture of what happens. So phage predation, you can imagine that it has two steps into it. One is how quickly a phage kills a bacteria. The second step is how quickly that bacteria that was killed and releases new phages go on to infect other bacteria. Now, these processes, because of this sort of infection and follow-up reinfection of other population, is a exponential process. And therefore, if there is a heterogeneity in the sort of timing of lysis and reinfection, for example, you get nonlinear advantages. So therefore, if the distributions becomes wider and wider, the population with a shorter lysis time actually outcompensates for the populations or overcompensate for the population with a very long lysis time. So in a simplistic scenario, if different features of the infection, like how many phages are released per unit time, are not having trade-offs with each other, you'd expect that a bacterial population with higher diversity will actually be eliminated faster. But there are these trade-offs.
that of course, as the cell can be, when the cell becomes lysed very quickly, it often produces too few phages to reinfect. So these trade-offs shape the sort of answer. So in some spectrum, definitely additional extra heterogeneity helps, but only up to a certain point. After that, it starts to hurt. I'm talking
Wonderful.
in the context of the phages, so it makes their process efficient. Yeah.
Wonderful. All right. Thank you, Dr. Bakshi, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Molecular Machines and Industries, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.
Thanks, everyone. And thank you for the invitation. And thanks, everyone, for your attention and good questions. Thanks. Cheers.